Because it depends which one you want to put in front of the other.
I actually understood this at one time, but I don't, because we were trying to model it for them. So the SINs come up at the decline
Of the prochlorococcus, yeah. Or before.
Yeah.
We can't tell which, right?
Yeah, they come up before. They come up, so see the thermocline is moving at that white line up there?
Yeah.
All right. So as the thermocline is coming up, you see the SINs come up, right? And then they decline off, and as because they probably just use up all that nutrients, so there's probably no free nutrients in the water at that point, and then pros take over, right? And you get this prochlorococcus system. This is also where the heterotrophs take off, by the way. So as the SINs die off and the pros come in, the heterotrophs start chewing away at the system too. And so we may be seeing a heterotrophic community, even though we're correlating with the prochlorococcus. Is I think how that story goes.
Do you know if any of this stuff was disturbed dramatically by hurricanes?
No. Yeah, you don't have to worry about it out there, right? You don't see those sorts of mixings. You could get it to happen, but you can watch the thermocline doing what it's supposed to do. It's just that typical cycle out there. It's not usually hurricanes that turn over the ocean, by the way. It's actually usually internal waves that are doing it, and then internal waves will, there's times when you can look at the Pacific and you can watch an internal wave running across it, and you can see the bloom following the big wave, right? All right, so these are the genes associated with transduction in the human system, okay? And so this is a different one. So I mentioned at the beginning that we've had cholera, and then basically there was CTX, the gene that, so if you just give people protein, the CTX protein, they develop the symptomology of cholera, right? So it only takes out one protein to do it. And that's actually true of all of these things for the most part. You don't really need the bacteria, you just need the toxin itself, okay? And that toxin is moving on phage and plasmids, right? So the way that you build a new pathogen is you put it on a mobile element and you stick it into the right background for it to be able to do it, okay? And so you can imagine in the marine environment, cholera, the Vibrio cholera, may actually be a pathogen or a symbiont on tons of different things. And all it's doing is switching in and out things like CTX or other phage-encoded things. All right, and this is to show you actually how common this thing is. So this is where we went looking for these genes in particular environments, around San Diego. And we do actually have, Shiga going on here. But we haven't had cholera in San Diego for a long time, basically since they drained the swamp, right? So cholera is not a disease that's going on in the city. If you go out and you look, you can actually find cholera toxin fairly consistently, at least in water samples, right? So literally you can find this like 50% of the time. And furthermore, we can show that they're in the viral fraction. So about 1% of the samples will actually have one of these very pathogen-specific sample, or specific genes in them. So they're really common out there. Based on this and some other stuff we know, we can estimate that any one of these encoded genes, or whether it's the Vibrio cholera gene or it's the things like polymerases and Hector and Paris, you'll actually see that there's somewhere at 10 to the 25 copies, which I showed you guys earlier, okay? So that what you see is these massive movement of genes out there. And we think nowadays that they're actually moving between phages because we've actually seen breakpoints, just like with the PSB, where we see it in different, cyanophages. If we look at the outside of these genes, we see that they're in different phages. Does that make sense? Okay. So these guys are mobile in the phage genomes, but not in the same phage genome. Yes, sir?
Has anyone looked at, like, when they pick up these exotoxin genes, how it's bad for the host?
Right.
Like SeroSwat, they would all have them, right?
Right, so these guys should be bad for the host no matter what. So the genes are actually on in this these things. They target things that actually all eukaryotic cells would have, right? So if you are carrying diphtheria, you can actually cause disease in anything from a an archaea to a single cell algae to a human, right?
Oh, no, I mean actually the
Yeah.
microbe that has it with the
Oh.
phage.
Yeah, so it's probably a bad idea from the microbe point of view in some ways, because really what it looks like, this isn't always true, but often these things are actually involved in only the lysis event, right? So what will happen is that the phage is turned, the bacteria, into something that turns a human into a bioreactor to make more phage, not to make more bacteria. All right, oh, sorry, I just said that. Okay, so the way to think about this is that, this brings different scenarios that we should think about. Basically, we're probably producing pathogens all the time, or these other recombinants out there. And the one that I think is most interesting is that you could imagine events where, of course, you have the non-human reservoirs. So this is like, CTX is famous for this because it's basically we can find it in birds, and we can find it in deer, and we can find it in humans, and we can find it in water, right? So it seems to be moving around a fair amount out there. The one that I think is kind of cool is the idea that you could actually eat the phage, right? And then it could infect a phage, a bacteria that's already symbiotic with you, okay? And turn it into a pathogen. And I don't know if that actually happens, but it's one of those models that I'd like to see actually happen.
Will you be testing this theory at any of your parties?
Here, have this cola. This one.
No, but in India they did this very nice story. I don't know if it's published, where they show that because of people, water is being recycled and at the same time not really well cleaned. What they find is they screen by PCR that compared to I don't know how many years ago, the individual carry not just one pathogen, but they carry a virus and a pathogen in this. And they were just showing PCR, but I wonder if it's what you were talking about. They didn't do that more, in detail. If it's a one bacterium or same bacterium who acquire the thing, but they totally see multiple carriage of thing, and they're like, cool, that's because you can do antibiotics or whatever. It's a virus, and then.
Yeah, I've seen a lot of
just
that
do
coming out
PCR,
lately.
yeah.
Where there's a ton of stuff of
Yeah.
these viral,
at the same time.
bacterial interaction things,
Yeah.
these polymicrobial
Yeah.
attacks on
Yeah.
things are,
Yeah.
there's definitely a lot of that.
Do you see any similar things in a positive point of view? So like probiotic sort of view, rather than
Yeah, there's no reason not to expect these sorts of behaviors happening in more of a commensal or mutualism, but there aren't that many examples of them out there.
Yeah.
That's probably just because of what people study,
Yeah.
right?
I think the trick for the human stuff too, from my point of view, is also not the mucus layer, but also the toll-like receptor that we have in our epithelium, because they're really not known. There's just one or two that have been described to recognize viruses, but it's probably they have many other receptors that would be able to differentiate or phage, or I haven't seen anything coming out. It's probably the receptors must be out there somehow.
That'd be really nice to know,
Yeah.
Natasha.
Oh.
Wouldn't it be nice to know? Yeah. Slap her. She's supposed to be doing something.
Waiting,
But no, she's going off getting pregnant, playing out, hanging out,
Hanging out.
We all know how that happens.
Tends to be the fashion in your lab,
This,
Okay.
this is going on the internet, by the
Oh,
way.
that's all right. So all of this and those things that I showed you about the toxin genes and moving around really brings you to this idea: our virus is actually just moving. So this the host range, right? So are they just moving between biomes? And this is Amiko Sana's work. And what she showed is this is that Wilcox ferment experiment, okay? And what we're going to do here, just like before, is that we're going to take and we're going to remove different things at different with different filter sets. And we're going to put them together, okay? So we're going to have, where we have 100 mils of DOM, and we're going to add a microbial fraction to it. Okay? And then we're going to watch it grow up. Okay? Or we've got 100 mils of the DOM plus the viruses, okay? And we're going to add the same inoculant. And then we're going to look over time, we're just going to count the number of virus-like particles and the number of bacteria that we find there. Does that all sound familiar? That's what we've done many times now. These experiments, right? So it's the same idea. And what will happen is that what's going to happen in this one? What's going to happen to the viruses?
It's going to rapidly increase.
No, they're going to disappear, right? Because there's not enough of them to support by mass action the viral population. So they disappear and the bacteria grow up to about an order of magnitude higher, right? Everybody remember that? Except for Jeremy.
Must be.
Well, he's Australian. It's not his fault. Okay. All right, so here's the phage per mil going along. This is time going along here. All right, so what happens is that if you don't have, so